Hi, everyone. Welcome to OsteoBites. Thank you so much for joining us today. My name is Christina Iptoma, and I am mom to OsteoAngel Dylan and Director of Scientific Programs at MIB Agents. And today on OsteoBites, we are joined by Dr. Isidro Cortés-Ciriano from EMBL's European Bioinformatics Institute. And he will discuss his work studying how ongoing chromothripsis underpins osteosarcoma genome complexity and clonal evolution. Thanks so much, Dr. Cortés-Ciriano, for joining us on OsteoBites today. Everyone, please feel free to submit questions during the presentation through the Q&A function in Zoom, and we will share those with Dr. Cortés-Ciriano after his presentation. A little bit more about our guest today. Dr. Cortés-Ciriano joined EMBL-EBI as a research group leader in June 2019 after completing postdoctoral training at Harvard Medical School under the supervision of Professor Peter Park and at the University of Cambridge under the supervision of Professor Andreas Bender. He completed his PhD at the Pasteur Institute in 2015 after obtaining MSc degrees in biology and biochemistry with a major in bioinformatics from the University of Navarra. The Cancer Genomics group at EMBL-EMI develops computational tools to characterize the mechanisms and consequences of genome instability processes in human cancers through the analysis of genome sequencing, data from clinical samples, and preclinical models using artificial intelligence and statistical methods. The ultimate goals are to deliver computational tools in the field of cancer genomics to study cancer evolution and facilitate the discovery of novel biomarkers and therapeutic targets, and analytical methods applicable in clinical settings to improve cancer diagnosis and treatment selection. Their research activities are articulated around three main areas: genomic instability and immune escape, early detection of cancer, and single-cell genomics. We're thrilled to have you on OsteoBites today, Dr. Cortés-Ciriano. Just a few announcements before we get started. We are now accepting abstract submissions for our Factor 26 Osteosarcoma Conference, which will be June 25th to 27th in Columbus, Ohio. Abstracts will be considered for oral and poster presentation, and I'll put a link in the chat for more info on that. Submissions are due on January 9th, and we are also offering a limited number of travel awards, and those applications will open in January. I did also want to share a call for submissions to a special journal issue of Discover Oncology, Making It Better for Osteosarcoma, Translation Research, Comparative Oncology and Beyond, edited by Dr. Alicia Valoria-Petit from the Ontario Veterinary College at the University of Guelph. This collection invites studies that aim to identify those key tools we are currently lacking to make it better for osteosarcoma patients. Translational comparative oncology research involving canine and human osteosarcoma is of particular interest, as well as studies at the interface of sociology, psychology, and oncology are welcome and encouraged. The submission deadline for that journal is May 4th, 2026, and I'll also put a link in the chat where you can find more info on that. And lastly, I just wanted to remind everyone about a fantastic resource. Turbo is a global virtual tumor board where top multidisciplinary osteosarcoma experts collaborate regularly. It provides a platform to discuss complex cases, share knowledge, and exchange best practices. And this research ensures clinicians worldwide can access specialist expertise. Physicians can submit cases, and researchers are welcome to attend and learn about real-world clinical challenges. Patients are welcome to ask their oncologist to present a case on their behalf. Turbo is hosted and sponsored by MIB Agents, and there's no cost to participate. Turbo meetings are bi-monthly, and our next meeting is January 14th. So I'll put a link in the chat where you can request a calendar invite and submit a case if interested. So let's go ahead and get started with our presentation today. Thank you so much, Dr. Cortés-Ciriano.
Thanks so much, Christina. Thanks, huge thanks for having me here. It's my pleasure, and I really encourage everyone to submit abstracts and attend the Factor 2026 conference. It is a great meeting, and it is really, really something special. So today I really want to walk you through some of our recent work on studying osteosarcoma evolution. And basically the question underpinning my presentation today and the work I'm going to present has been, how does osteosarcoma arise and evolve? So that's a question that basically we wanted to address here. So if we basically zoom out a bit and we consider what determines or what are the features of cancer, right? Already 100 years ago, a connection, a link between aberrations in chromosomes and cancer was established. And this is what we normally see under the microscope. So on the left-hand side, you have the karyotype, so the structure of the chromosomes for a normal cell. And one of the hallmarks of most cancers is that the chromosomes are highly scrambled, as shown on the in this example on the right-hand side. And these aberrations are caused by structural variants, or SVs, which refer to the deletion, amplification, or translocation of genomic segments in cancer cells. Thanks to whole genome sequencing studies, over the last 15 years, we've learned a lot about the patterns, types, and characteristics of structural variants in cancer genomes. And now we know that sometimes structural variants can occur in isolation, as illustrated in this example, where we have the diagrams of two chromosomes with a deletion and a duplication. And we can also represent these types of alterations with the type of diagram at the bottom, where we represent chromosomal coordinates on the x-axis. And on the y-axis, we represent the total number of copies of each genomic segment. So for instance, here, we can see that there has been a deletion, so that's why we have one copy instead of two. And the structural variants, so the adjacencies that are created or found in the cancer cells that are not present in the germline genome, are illustrated with these arcs. So sometimes these types of events happen, right? So a normal chromosome with some isolated events. However, the elephant in the room is that in most cancer genomes, right, with the structural variants, the majority of SVs map to so-called complex genomic rearrangements, or CGRs, which span from 1 to over 15, 18 chromosomes, you'll see more later, and involve tens, hundreds, sometimes thousands of structural variants. So over the last years, my group and others have, we've collectively shown that these type of patterns are very pervasive across diverse pediatric and adult cancers. They are very heterogeneous within and across cancer types. They are mechanistically linked with drug resistance and predict poor prognosis, and are involved, for example, in amplification of the oncogenes. And we'll discuss more about that in this presentation. And just to give you like a notion of how important and pervasive these complex events are, these are results from the pan-cancer analysis of whole genomes project, in which over 1,000 researchers analyzed the genomes of over 2,600 tumors. And here, each panel represents a cancer type, and the y-axis quantifies the number of structural variants that fall of a particular type, right? So in essence, all of these colorful violin plots here represent those simple structural variants, and the gray violin plot basically accounts or represents the number of structural variants mapping to these very, very complex crazy chromosomes. And you can see that across very diverse cancer types, including soft tissue liposarcoma, ovarian cancer, osteosarcoma here, breast, brain, etc. The gray violin plot is the one with the highest number, right? So across diverse cancer types, we see this phenomenon that structural variants are clustered and affect large regions of the genome. So the question arises, what mechanisms underpin the formation of these complex events? How do they arise, right? Back in 2011, there were several seminal papers presented, especially one where a pattern like the one shown on the right-hand side was discovered. And this was kind of quite surprising, right? Because you can see, we can't forget about the blue line now, which represents the copy number of the least amplified allele or parental chromosome. So just focus on the black one. And we can see that there is an oscillation pattern in the context of a cluster of structural variants. So how can this arise, right? So the authors at the time performed computational simulations to basically test the hypothesis that, well, this is a result of the gradual accumulation of structural variants. So in the computer, one can add one structural variant at a time, right, of a particular type, a deletion, amplification, or a translocation. But when they did the simulations, really the result, right, of the simulations didn't match this pattern, right, the observed pattern in the cancer genomes. However, when they consider the possibility that all the rearrangements could have been acquired in a single cell division all at once, the simulated data really matched the observed patterns in the cancer genomes. And this led the researchers at the Sanger Institute to propose a new mechanism called chromothripsis, which means shattering of the genome. And they proposed that the chromosome undergoes fragmentation into tens to hundreds of fragments, some of which are lost to the cells, and the others are stitched back together in random ordinal orientation, creating a derivative chromosome with many rearrangements, as it is shown on the right-hand side. And something important I want to highlight here is that the computational analysis here allowed to the research to identify new patterns and propose hypotheses about how these patterns might have been acquired, and only then experimental work identified the mechanism. So over the last few years, there has been pioneering work by David Pellman and other groups looking at basically trying to investigate the mechanisms underpinning this complexity. And now we know that these patterns of chromothripsis can arise through the fragmentation of chromosomes, entire chromosome or acentric fragments in micronuclei, as well as through the resolution of a dicentric chromosomes that result in an anaphase bridge, and their mitosis they are resolved, generating also these patterns. This particular mechanism, chromothripsis, is something that I studied quite in detail during my postdoc with Peter Park and more recently in my own laboratory. And initially, chromothripsis was estimated to happen in 3 to 5% of tumors, and these estimates came from the analysis of SNP array data, which is one of the early technologies that was used to analyze copy number alterations in cancer genomes. But that technology, cannot detect rearrangements. So the estimates were very low, because with whole genome data, we estimated that around 40% of all tumors have chromothripsis, and in some cancer types, the frequency is very, very high. And here I'm marking osteosarcoma, which is basically showing that in almost all tumors, we observe complex chromothripsis patterns. Chromothripsis is one of the mechanisms that can generate this genomic complexity, but since 2011, other mechanisms have been identified, such as chromoplexy that underpins the formation of gene fusions, as well as chromoanasynthesis, which is a copy and paste mechanism whereby gains of or copies of genomic regions are accumulated and stitched in tandem. But the key challenge, now going directly to osteosarcoma, is that the patterns that we see in osteosarcoma genomes cannot be explained by these non-rearrangement mechanisms. And in most pan-cancer studies genomics studies at the pan-cancer level, osteosarcomas have always been a bit of sidelined, because the number of samples was always limited, so not high enough to make strong conclusions. And also because the patterns that are observed in these cancer genomes are extremely complex. And here on the right-hand side, I'm just showing you one example. In this type of representation, each of these boxes is a chromosome. We represent the set of chromosomes in the human genome as a circle. And the lines in the middle are like the lines in the previous plots, are basically represent adjacencies, translocations, or connections, links between genomic regions that are specific to the cancer cells. And this is like a hairball. This is thousands of rearrangements involving many chromosomes. And sometimes we see 15, 20 chromosomes involved. And when we look at the karyotypes, that's just the example on the left-hand side, you can see that the chromosomes are, we should see basically 2 of each type here, right? And we can see that it's all over the place. So how does this happen, right? Just to give you going back to a type of representation that I have introduced at the beginning that I will be using throughout the talk. These are real examples of osteosarcoma genomes, where at the top you can see one of these complex arrangements involving 3 chromosomes. So again, chromosomal coordinates on the x-axis and the total number of copies represented on the y-axis by these black bars. And the rearrangements, right, these new connections between genomic regions represented by these lines. You can see massive complexity, amplifications. Note that we cap the copy number at 10, but sometimes these amplifications can go to much higher numbers. So this means that we have many copies of a given genomic region. But trust me that the one at the top is one of the not so complex examples, right? Often we see cases that involve 15, 18, 20 chromosomes, and they are so complex that we cannot even render in the computer. So at the bottom, just to make the point, I've brought one of these cases, right, involving many chromosomes, the only one that we could render. But this is not the most complex ones by far. So at this point, right, there have been studies of osteosarcoma, whole genome sequencing studies, and we couldn't still really figure out how these rearrangements happen. So the question arises, what are the technologies and experimental designs that we need to really address this question of how does this complexity of osteosarcoma arise? And now I'm going to walk you through the experimental design that we've implemented to address this question. And I would like to, at this point already, kind of mention that the work I'm going to present has been a fantastic collaboration with Professor Adrian Flanagan from University College London and the Royal National Orthopaedic Hospital in London as well, with whom we've been working in the analysis of various sarcoma types, but with a strong focus on osteosarcoma. And this work was led by Jose in my team, now PI at the Bottom Champalimot Center in Lisbon, and and pathologist training with Adrian. And this is another good opportunity to mention that collaboration between computational labs, clinicians, and pathologists is essential to make progress in the complex challenge of understanding diseases such as osteosarcoma. So in essence, the experimental design that we propose is the notion is the following. For each tumor sampled, we isolate different macroscopically distant regions, and we perform whole genome sequencing on each of these regions. Then we identify somatic alterations, so mutations, right, changes in the DNA in each of these regions that we use as genetic barcodes to trace the phylogenetic tree, right, or the genealogy of the different regions or clones in the tumor. And with these trees, we can do many things, right? So we can assess how diverse, how different the different clones in a cancer are, but we can also order the mutations as they are acquired throughout tumor evolution. So in other words, we can study tumor evolution retrospectively in humans using clinical samples, right? And in the specific context of osteosarcoma, this also allows us to understand how the cancer genome breaks and rearranges over time as the cancer cells grow and expand. So to do this work, we performed multi-region sequencing on 30 plus osteosarcoma specimens, but we also analyzed a lot of newly generated single region whole genome data from osteosarcomas, as well as virtually all published osteosarcoma data, assembling the largest collection of osteosarcoma genomes to date, uniformly processed and analyzed. And the first question we asked was, well, now that we have many more samples, we have more statistical power to identify genes that might be mutated frequently. Well, is there any gene mediating osteosarcoma development that we can now identify that was not in the past? And the answer is no, right? So the most recurrent genes that we found with mutations were the classical drivers in osteosarcoma, p53, of course, RB1, CDKN2A, ATRX. So nothing really novel on that front. But we did observe that chromothripsis rates were very high, as well as the rates of whole genome doubling, or WGD, as abbreviated here. So now let's move to the multi-region data. So here in this diagram, each row represents a chromosome, and each column represents a patient on which we performed multi-region whole genome sequencing in their tumors. And in essence, every time you see a square, a red or a blue square, it represents that some of the chromosomes were heavily rearranged in some of the in one or more, but not all of the tumor regions in the tumor, which is something that we were not expecting, and we found that to happen in 74% of basically 3/4 of osteosarcoma genomes. And I'm going to show you today, but we also analyzed, motivated by this finding, other cancer types, and we also observed that this is frequently detected in other cancer types, which is something very novel that has important implications for researching chromothripsis, osteosarcoma, and other cancers as well. So just to give you an example, this is one specimen on which we performed multi-region sequencing on 6 regions. One is not labeled on the specimen because that was the biopsy sample. And in this type of plots, we represent, again, the genome as a circle. So it's of these sectors here represents a chromosome in increasing order. And in essence, the innermost circle represents the structural variant. So the rearranges the connections between different chromosomes that in turn represents how the structure of the chromosomes is in a particular region. And what you can see straight away is that these plots look very, very different. And this means that the structure of the chromosomes is very, very different across regions that are just centimeters apart in a given tumor. As I was introducing, we did this across various sarcoma subtypes, and we also found high rates of this subclonal chromothripsis, or chromothripsis complex rearrangements happening in a subset of the tumor regions. But with this data, we could also study something very interesting for the first time. So now we are seeing that there are many, sometimes hundreds of rearrangements that are happening just in a subset of the cancer cells in a tumor. So now we can try to basically address the following question. Well, where are they acquired? Do they fall in specific regions in the chromosomes as cancer cells grow, or where do they happen? So I'm going to show you one example that is not anecdotal. This is a representative of what we observed across the entire cohort. So in this example, I'm showing you here three tumor, basically the copy number pattern, right? The type of flow that I've been using throughout this talk were for chromosome 18 across three tumor regions from the same tumor, right? So again, the y-axis represents the total copy number, and we are showing here the data that has been calculated using the data from each specific region, and the lines represent the structural rearrangements that we estimated to be acquired very early during tumor evolution based on their presence in all the regions, right? So these rearrangements were detected in all the tumor regions, which means that they were acquired early during tumor evolution. But now, if we also display the rearrangements that are specific to each region, what we can see is that there are hundreds of rearrangements accumulated in each of these regions that are specific to a single region or two. But something very interesting is that they map exquisitely to the Q arm, or basically the region towards the right-hand side of the chromosomes. But the part on the left, which is what we call the P arm, is unaffected. So in summary, what we found is that these rearrangements that happen early prime the cancer genome for ongoing genomic evolution, for the acquisition of mutations throughout cancer evolution. So at this point, we were seeing, I've mentioned the word rearrangement already many times, right? So far. But the question we had is, okay, well, they're happening across the genome, but are there any regions that are affected more often than not, right? And is there any recurrence at the level of where these rearrangements are being acquired? And it was a bit, underwhelming in a way that we found that the only region that was statistically significant when we did this recurrence test. So basically what we compute is what is the expected, right, number of rearrangements that we would expect to find at a given genomic region based on some of the characteristics of the genome at that location, and then we compare what we observe. And the only region that stood out was the 53 locus, right? So when we looked at basically how 53 and chromosome 17, where 53 is rearranged, we've been talking about chromothripsis a lot. We would expect to see patterns like this one, which is what we call the canonical chromothripsis pattern, where we see the characteristic copy number oscillation pattern with hundreds of rearrangements. We only found one case with such pattern, right, across over 250 cases. So that was very surprising. So how is chromosome 17 rearranged in osteosarcoma? So we did a simple yet extremely powerful analysis, in my opinion, which is the following. On the x-axis here, I'm showing chromosomal coordinates for chromosome 17. And on the y-axis, I'm showing you the fraction of tumors, either pediatric or adult, that show deletions, right, of the chromosome. That's the blue line. Rearrangements, basically, where cuts, let's say, double-strand breaks in the genome, or amplifications, and that's a red line. And very surprisingly, you can see that the two plots, right, for pediatric and adults, are very similar to each other, and we can see that around 50% of cases have this peak of rearrangements at 53, indicating that there is a recurrence of rearrangement at that location, and that over three-quarters roughly have deletions of the terminal part of chromosome 17. Well, interesting, but how does this happen in other cancers? So we repeated the same analysis using data from over 30 types of more common cancers, right, from the pan-cancer analysis of whole genomes project. And we did observe a very different pattern. In this case, we see that the chromosome arm with 53 is lost in about half of the cases, but yet the pattern is very different. In this analysis, the problem is that we are putting together many cancer types, the biology of which is different. So any pattern that happens infrequently, right, in cancers might be diluted, as could be the case with osteosarcoma. So we repeated the analysis focusing on specific cancer types, and we paid particular attention to those that are characterized by recurring mutations in p53, as is the case in osteosarcoma. And here, for ovarian adenocarcinoma, you can see that almost all the tumors show a loss of chromosome 17, indicated by the blue line. But yet again, the pattern is different. And then we analyzed esophageal adenocarcinoma, which is a cancer type also driven by p53 inactivation and with high rates of chromothripsis, whole genome doubling, extrachromosomal DNA, and the types of alterations that we see frequently in osteosarcoma. And here we see that 3/4 of the tumors have loss of the chromosome arm with p53. But once again, the pattern is very different. So long story short, this pattern that we observe in osteosarcoma is something that we only detected in this cancer type. And we looked at basically over 30 types of adult cancers as well as pediatrics. So this pattern seems to be something very specific to osteosarcoma, as I will discuss more in a second. So at this point, after having looked at many plots, right, like this one, what we call in the lab manual genomics, it's not all AI. You really need to get into the data and look at the raw data. We started to observe a pattern like this one, which is what the previous plots represented. So in essence, we can see that there is loss of this terminal part of chromosome 17 at this location. That's indicated by the blue line here. Downstream p53, we see amplifications. And those amplifications are connected to other chromosomes where we often see some regions, small regions amplified containing oncogenes, such as in this case CCNE1. So this is a representative example of the pattern that we were summarizing at the cohort level in the previous plot. So when something interesting is when we looked at where the p53 is being rearranged or cut, as it were, right, where the chromosome is being affected by this rearrangement, we observed that in half of the cases, roughly, the breakpoints were mapped to intron 1 of the gene. And this is very interesting because inactivation of p53 could have been achieved, right, had the breakpoint happened in other regions of the gene. So we don't know why this is the case, but it's an observation that is very, very important. And then in the other half of the cases, roughly, the rearrangement happened downstream p53, leading to the loss of the entire or one entire copy of the gene. And we can also, with sequencing data, what we can do is analyze how much of the signal that we detected or the sequencing data or sequencing reads in this case, right, the readout that we get when we perform sequencing, how much of the readout supports the mutations that we see, right? And this is what we call the allele fraction. And this basically is a proxy for us to understand how many of the cancer cells have a particular alteration. And what we observed, which is summarized here in this diagram on the right-hand side, is that most of the cancer cells, if not all, had this rearrangement in p53, but all the other rearrangements were present in a smaller fraction of the cancer cells, as indicated by the allele fraction being lower. So in other words, what this data indicates is that the rearrangement in p53 is one of the early events in the evolution of these cancers. But at this point, many of our colleagues, when we're discussing this data, would come back to the following question. Well, we see this complexity. This is another example, right, of an osteosarcoma genome, so in the alterations in p53. How many chromosomes are altered at this location? How is the cancer genome structure? Right? This is very complex to be understood with this type of diagrams. So most of the in this particular project, we used a lot of short-read whole genome sequencing data, and this is the dominant technology that's out there for analyzing cancer genomes, and that is being also used in the clinic. And I'm going to show an analogy to make a point. The readout that we get with this short-read sequencing are basically, as it were, sentences of 150 letters, right? So we can think of the genome as a string of letters, amino acids, and we got that. Nucleotides, amino acids are the proteins, sorry, nucleotides. It's late over here. And with this equivalent to, and then we have the reference genome, right, that we have assembled the entire sequence of the genome. And what we need to do with these sequence rates is map them, right, understand from which region of the genome they are coming from. And one of the challenges in human genomics is that there are many regions of the genome that are repeated along the way. So when you have a readout of 150 base pairs, there are many regions on which or to which you could map the read equally well, right? And so to use an analogy, so reconstructing the structure of the cancer genome is like trying to reconstruct the puzzle of this little bird. So if you have a piece showing the beak, right? However small that piece is, you know where it's going to go, right? Because there's only one beak in the picture. However, you have like a picture that's all green, right? The color of the sky. You don't know where to place it because there are many locations in this little picture where the small green piece would basically go with the same accuracy, right? Or with the same probability. So we cannot basically place or align, as we call the sequence reads. However, there is a new set of emerging technologies that we call long-read sequencing that allow us to not only read 150 base pairs, but much longer, sometimes tens of kilobases. And in essence, this facilitates our analysis because following the analogy of the puzzle, the task now is to complete the puzzle, but using much bigger pieces. And in this case, there is no ambiguity. We know very well how to put the pieces together, right? Or in other words, how to reconstruct the structure of the cancer genome. So for the first time, we performed long-read sequencing on the Osteosarcoma specimens, and this allowed us to address the question of how is the cancer genome rearranged. So as it often happens with novel technologies, I mean, there is a lot of potential, but also many challenges. And what in the case of long-read sequencing, there are many errors in the readouts, right? Like typos, as it were, in the sentences that we read. And so Caroline and Hilary in the team developed an algorithm called Savannah to detect these copy number alterations, the number of copies of each genomic region that we see using long reads, as well as the structural variants or rearrangements. And the key innovation here is that we used machine learning to distinguish the true signal corresponding to biological alterations from the sequencing errors, right, or the typos. And this allowed us to basically detect the rearrangements with high precision and reduce the number of false positives massively as compared to existing tools. And why this matters, is that now we can reconstruct very complex aberrations, such as the ones shown here, using these novel technologies with the same accuracy as with standard technologies used in the clinic, such as Illumina whole genome sequencing or short-read sequencing. And this is an example where we basically performed short and long-read sequencing on the same tumor, and you can see that the two plots are basically identical, meaning that we can now derive the important biological signal using these emerging technologies. So going back to the sarcoma, we have this question of what is the chromosomal configuration, right? The structure of the chromosomes in the tumor with this type of alteration. And so in the team, what she did was to basically put together the puzzle, right, of the different parental copies of the chromosomes in osteosarcomas. And this is just an example corresponding to the case shown on the screen at the top. And here we can see that when we made the puzzle for one of the parental alleles, we saw that it was massively rearranged, but the other one was not. And what this is telling us is that all the complexity that we see at the top is only affecting one chromosomal copy, which is an important piece of information to understand how these chromosomes evolve. So now, if we put all the evidence that I've been walking you through together, that allowed us to identify a mechanism that we termed loss translocation chromothripsis or LT chromothripsis that we found to drive the evolution of around 50% of high-grade osteosarcomas. And what happens is the following. In a cell in which one of the copies of p53 has already been inactivated, and this can be an inherited mutation, as in the case of Li-Fraumeni syndrome, or a mutation acquired during life. The other copy of chromosome 17 that has a functional p53 copy undergoes a double-strand break at the locus of p53, as I have been showing you, in intron 1 or downstream the gene. And at this point, that leads to the biallelic or complete inactivation of p53. This double-strand break leads to the formation of what we call an unprotected chromosomal end. So this is a chromosome that doesn't have the tip, right? So this makes it unstable. And what we found is that these chromosomes translocate to a second chromosome, shown in yellow, near an oncogene. This translocation generates a dicentric chromosome, basically a chromosome that has two centromeres, which are the regions of the genome that are used as anchors to pull the chromosomes apart during cell division. And this is illustrated by these small circles here with a lighter color. And what we know is that these structures are very, very unstable during mitosis, during cell division, and they are fragmented. And so what happens here is that this dicentric chromosomal structure will be resolved, right? They will undergo a double strand break in the next cell division. So these steps, these alterations are at what happens in the cell that gives rise to the entire osteosarcoma, right? Or what we call the clonal alterations, or what they happen in the most recent common ancestor, as we say. And interestingly, what we observe is that during this process, which is basically two cell divisions, half of the cases acquire whole genome doubling. So the entire complement of chromosomes is doubled. At this point, the chromosome 17 here, with a bit of chromosome of the other chromosome, with a yellow chromosome with one copy of the oncogene, can undergo various or can take various routes, right, or trajectories, as it were. It can fuse to a third chromosome, shown in purple, and we observe that this happens also, again, usually near an oncogene, creating a dicentric chromosome that, once again, in the next cell division, will undergo fragmentation. And at this point, we are again at the square one because a chromosomal copy without the tip, right, is created, unstable. We are back here. Or what can happen is that this chromosome, upon replication, so the DNA is copied before every cell division, will fuse to its sister chromatid. And this will again create a dicentric chromosome that will be unstable, will be fragmented, and then we're back at the square one. So what we observe is that this process is repeated on and on. And this creates intratumor heterogeneity, or basically this leads to different groups of cells that are very different at genomic level, although they are basically cousins or sisters to each other. And during this process, many chromosomes can be involved, sometimes up to 20 chromosomes, generating at the end these very, very, very complex chromosomes composed of fragments, pieces, sometimes copied to high copy number levels from many, many chromosomes. And in this process is we observe that extrachromosomal circular DNA, or basically circles of DNA with oncogenic copies that can be amplified to very high copy numbers and drive tumor evolution, are also formed. And this is the process that we discovered to basically drive the evolution of about half of high-grade osteosarcoma cases. And just to give you an idea of the relevance of this process, so what I've been introducing this concept of phylogenies or genealogies of the different cancer clones or different cancer cells. And we can basically perform this analysis using point mutations. So those are basically one letter or one nucleotide mutations in the genome, or with the structural variants. And this is an example of an osteosarcoma in a leaf from an individual. And the key point I want you to show you is, or I want to tell you something that is based on the observation from this sector of the phylogenetic tree. We can see that these branches, which represent the number of mutations in different regions of the tumor, emerge from this flat line. This looks very much like the phylogenetic trees for embryos. What this is telling us is that there is massive clonal diversification in a few cell divisions. And we know this because in every cell division, mutations are accumulated. But here, these branches do not share mutations. And this indicates that there is massive diversification from early on, indicating that this process of LK chromothripsis drives rapid clonal diversification and rapid growth, which is also something that is consistent with clinical practice of how osteosarcoma evolves. At this point, thanks to the very diligent efforts of Professor Adrian Flanagan and her team, one of the key features of this dataset is that we had very, very detailed clinical annotations for most patients. So with the clinical annotations and so much genomics data, we asked the question, what are the genomic alterations that predict prognosis? And this was very important because this was the first time when we could do this at a scale, right? And we looked at the correlation between prognosis and many, many, many genomic features, right? Some of which are shown here in this plot. But the only one that really showed predictive power was the degree of loss of heterozygosity. Or in other words, this means how many chromosomes or chromosome pieces have been lost during tumor evolution. And this is something that we found to be to hold even when we corrected for non-confounders or survival, such as metastasis diagnosis and other factors. And it's important because tools to prognosticate or predict the prognosis of patients are very, very much needed in clinical practice for osteosarcoma. And we found this to be highly reliable, but of course, other validations using other cohorts are required to make more conclusive statements. So in summary, hopefully I will have convinced you that analyzing multiple regions of the same tumor using whole genome sequencing allows us to study tumor evolution retrospectively in humans. And in this particular study, we discovered the Jones translocation amplification chromothripsis mechanism, which underpins the evolution of about 50% of high-grade osteosarcomas, and genome-wide LOH, or in other words, the fraction of the genome that has undergone losses from chromosomal copies is a strong predictor of prognosis for high-grade osteosarcoma. Really, the key people to highlight again are, in this case, Professor Adrian Flanagan, our fantastic collaborator, as well as the two leads of the study, Jose and Solange. Thanks, of course, to all the participants, the patients and their families who donated samples to the study, without whom this would be impossible. There were many people involved in the study. I'm listing them. They're here using the solution from the publication. Many funders, you can find them there. We're most thankful to all of them. And yeah, thanks so much for having me here. And I'm most happy to discuss. And yeah, it's really cool to be here. Thank you so much.
Thank you so much, Dr. Cortés-Ciriano. And again, we're so happy to have you here on OsteoBites. I know this paper, and I put the link to the paper in the chat for people to be able to easily access. But I know that came out in January, so great to kind of have you here to kind of help walk us through that, because I think it actually, I think your work is referenced a lot, I think, to kind of identifying potential vulnerabilities for targeting osteosarcoma. So great to have you kind of explain it on Zoom for us. So everyone, please feel free to add questions to the Q&A in Zoom. And in fact, I think we have one that just came in. Okay, first question: can whole genome LOH be identified with clinical available molecular genomic diagnostic tools?
Yes, with the SNP arrays. Actually, when we identified LOH as a predictor of prognosis, we found a very nice paper from 2013, I think it was, where they had already identified LOH as a potential biomarker. Prognosticator for osteosarcoma. So yes, SNP arrays can be used to infer LOH. We need to basically assess in larger studies whether the resolution afforded by SNPs or SNP arrays would be enough to give accurate predictions, but from our initial comparisons, we think that is the case, because the losses in these cases are big enough to be detected reliably with SNP arrays.
And just to make sure I understand that, so is it a biomarker that would be easy to test for today? So if a patient walked into the clinic today and wanted to test for that, is it something that an institution could easily do, or does it require kind of this more high-depth, multi-region whole genome sequencing?
No, it could be done basically on a single region, even a biopsy, right? So SNP arrays are a technology that's pretty old already, right? So it's well established. I would also like to say that whole genome sequencing is becoming more affordable, and whole genome sequencing is a very simple experiment, right? One needs to extract the DNA and sequence it, right? Obviously, the cost has been a blocking factor so far. And the complexity of the analysis, the data storage, the qualified bioinformaticians that are required to do it. But there are many advances. For example, in the UK, in the Netherlands, some institutions in the US are implementing whole genome sequencing, and that's clearly the future. So as of today, SNP arrays could detect LOH, but moving forward, whole genome sequencing will become more common. But I insist that more work and more additional validation using other cohorts is going to be essential to identify the power of this marker and any others to really assess their power. And this is again another opportunity to highlight the importance of collaboration to study and to make progress in diseases that are not so common, like osteosarcoma.
Great, thank you. Okay, question from Sasha Blay from the Schlein Lab in Toronto. Thank you. And to what degree do you believe these chromosomal and CN changes set the stage for later chemo resistance, and what mechanisms are most useful for the cell to overcome therapeutic pressure?
Yeah, so it depends on what therapeutic pressure. So, for example, the ecDNA elements and the capability of the tumors to generate ecDNA elements is a known mechanism that has been established of drug resistance that is very common, for example, in glioblastoma. We didn't see much evidence of that here, but the type of samples that we had were also not the best to address that question because we didn't have longitudinal samples. In terms of chemo resistance, something that I didn't show today is that with the multi-region data, we could study which regions had basically acquired mutations associated to the treatment. So in other words, where they had been exposed to the chemotherapy. And what we found is several patterns. So in some cases, we did find that some regions had not been hit by chemotherapy. And this can be, can be due to various factors. But in other cases, we did see that the clones that basically led to resistance had been hit by the chemotherapy, but had not died, right? So this again points to our lack of understanding of the mechanisms driving drug resistance in osteosarcoma, and we need really longitudinal cases to address these questions.
So good segue to, I know in your sample set, I think you did have, I think it was like 40% of samples were treated with MAP. And I was wondering how the chemotherapy affects ongoing chromothripsis. And because I actually first learned about chemo leaving these mutational signatures actually from a presentation Sasha did. But kind of curious just now that we know that there's this kind of ongoing chromothripsis and evolution, how might chemotherapy affect that? Because that's kind of, patients, as soon as they're diagnosed, they get that immediately.
Yeah, we don't know. That's the honest question. We don't know. And also one of the challenges studying these alterations, chromothripsis, rearrangements, etc., is that they are harder to detect than point mutations or basically the traces that chemotherapy leaves in the genome, right? So we don't know what happens. What happens in basically a small set of cells is something that we cannot detect at present unless we do sequencing on small groups of cells. That's something we are doing now at the moment, but the results are not mature enough to be shared. Hopefully at Factor next year.
Awesome. Okay, a couple more questions coming in. So some osteosarcoma occurs in patients with cancer predisposition syndromes beyond Li-Fraumeni syndrome, like RB1 and RECQ4L, or some osteosarcoma samples do not have P53 mutations. Is LTA seen in those specimens?
So LTA, implies the biallelic inactivation of P53. So in all cases with LTA, we see the biallelic inactivation of P53. And in terms of samples from cancer predisposition or individuals with mutations in cancer predisposition genes, we only had a few. I think it was like 5 or 6 LFS cases, but not others. So to be determined.
Is there the potential to assess these CGRs on the single cell level, or is there just not enough DNA for current single cell technologies to get adequate reads to call SVs?
That is my dream. So for years, single cell genome sequencing has been not mature enough because basically the readout that we got was quite not good enough for detecting rearrangements, right? It was okay to detect some gross copy number aberrations or point mutations with limitations. And that would be the ideal experiment, right? To sequence the entire genome of a cell, many cells in a tumor, and reconcile the phylogenetic to a single cell level. Now there have been improvements over recent years in single cell technologies, and that coupled with the decrease in sequencing cost, leaves me hopeful that soon, in a number of years, we'll be able to perform exactly that experiment. I'm very much looking forward to to see what we can learn from that.
Great. Question about what do you think about Brocanis HRD in osteosarcoma? Can it be a therapeutic target in your opinion?
Yeah, thanks for this question because it's usually like a recurrent topic. So in this cohort, there's again 350 plus tumors. The sequencing depth that we had was in most cases over 100x, right? So basically, this means reading the genome more than 100 times, right? For its case. And what this means in practice is that we had very, very good sensitivity, to detect these rearrangements and basically the mutations that can be used to predict whether homologous reproductive deficiency is indeed something happening in osteosarcoma. And to predict basically HRD or homologous reproductive deficiency, there are several tools that can be used to make the prediction based on the mutation detected in a cancer genome. So we used basically state-of-the-art methods. We didn't find a single case. That doesn't mean that is not happening in sarcoma. It could be at the subclonal level, but we didn't detect the patterns or the features of HRD. That's something that's described in the paper. We did not. That's what I can comment, whether there are some subclonal alterations or groups of cells developed sensitivity to basically or HRD features. That's something to be determined. But in our case, we didn't see HRD.
So I know that I think about like 40% of your sample set were pediatric patients under 18. And I'm wondering if you saw a difference, if you looked at age and if that's a factor at all. So for example, these 50% of osteosarcomas that are driven by LTA, chromothripsis, is it kind of same across different age groups, or do you see any difference in?
Yeah, well, we saw roughly the same frequency of LTA across age groups. Yeah.
Gotcha. So I know you're talking about kind of these different, you're seeing these different subclonal changes within the same tumor, different regions of the same tumor. But I'm curious, because I think you might have also had some metastatic samples as well in the data set of kind of what's the role of chromothripsis in driving metastasis?
We are investigating that right now with the Cancer Lab at St. Jude's and also with Professor Adrian Flanagan, of course, using different techniques. We didn't have many metastatic samples in this cohort. They were primarily primary. We had a few from the kids' first data set, but the quality of the sequencing data was lower than what we had. So that's a key question. What drives metastasis, right? In a few cases for which we had multiple tumor samples, including mets, we did see that there was what seemed a recurrent pattern, specifically that the clones that or the mutations that were found in the metastatic clones were acquired early, and clonal diversification happened early during tumor evolution. And that will be consistent with the presence of oligometastatic disease in some plant-based diagnosis. But that is the key question, right? What are the factors or the features that make some clones capable of metastasizing? We are studying that, and we are very much into addressing that question. So if someone is interested in discussing or collaborating, please do reach out, because that is, I think, a key piece of information we need to all understand to make progress.
Okay, great. Couple follow-up question on that last one. In the multi-region cases, were the therapy-associated mutations unique or shared across multiple sites? Thinking about clonal evolution and timing of divergence.
The mutation associated with chemo, they were different across regions primarily. Yeah, in some cases we did observe basically mutations in the trunk of the phylogenetic tree, or basically the same chemotherapy-associated mutations in all the regions we analyzed, so indicating that they were acquired before there was diversification of clones, right? After treatment, we found a few of such cases, and that might indicate that one clone basically takes over. So the chemotherapy hits many clones, kills some others basically, or one in particular gets mutations from the chemo, but is not killed, and then it grows, basically, it takes over, right? It outgrows the others. We did see that pattern, but why is something that we don't know. And based on whole genome data only, we cannot answer that question. We can only see what has happened in terms of by looking at the trace of mutations, but what are the pathways or the which genes are activated, which features of the tumor microenvironment might be permitting the outgrowth of a clone-resistant treatment is something that, is to be determined.
Thank you. Another question building off the RB1 RECU4L question. While LTA is specific to P53, have you found any other sets of genes involved in similar patterns of TSG disruption coupled to oncogene amplification? Is it possibly in other cancer types with high rates of chromothripsis?
Thanks for the question, because that is an analysis that was required by one of the reviewers, and we did that across over 5,000 whole genomes from TCGA, spanning more than 30 cancer types. We did not find a single example of an LTA-like chromothripsis process involving a gene other than P53 and any oncogene. An LTA per se involving P53 and a given any oncogene, right, is something that we only found in osteosarcoma, and some LTA-like patterns in some mixed sarcomas, but at very, very low rates. And they were not as strong as in osteosarcoma. That's why we claim that this pattern is specific to osteosarcoma.
Great, thank you. So we're almost out of time, and I know you've kind of referenced a little bit of kind of some of the work to come, but can you maybe close us out with just this understanding this presence of the ongoing chromothripsis, kind of what targeting vulnerabilities that might suggest for us to kind of look into further, and also just kind of what's next, kind of on your next steps on the study.
Yeah, so I think that this work really illustrates that genomic instability is an ongoing process. So rather than targeting the result of that process, right, the amplification of oncogenes, which we have learned, unfortunately, that is common in various cancer types. For example, glioblastomas, some of the most aggressive cancers, right? And we know the cancer cells are very good at basically resisting treatments, targeting oncogenes that are amplified in chromosomal DNA or in the linear genome, right? So rather than targeting the end product of the instability, can we block the stability process itself? And that is something that there is very promising work from experiments in treatment-resistant melanoma and other cancer types. So that is kind of like an open question. What DNA repair pathways processes could we basically target to block this process? Whether that would, such a strategy would be toxic or to what extent is to be determined, but that is something important. The other kind of, I think, important conclusion from this work is that clearly the heterogeneity, which is what drives drug resistance in osteosarcoma, is something is a phenomenon that is acquired during the first cell divisions and persists during the growth of the tumor. So targeting oncogenes is not going to solve this problem. So can we think of other therapeutic avenues, right? Can we use immunotherapies to treat osteosarcoma? I think we really need to think out of the box and just abandon the classical targeted therapy approach for osteosarcoma. This is very strong evidence that there is a reservoir of genomic instability that the cancer cells can use. We need to think differently, right? And personally, I am very motivated about the potential of immunotherapies for osteosarcoma. They have not worked so far, but it doesn't mean that they cannot work, right? We need to understand why and see the potential. And that's something that we are exploring at present. So that's something that hopefully we'll be able to share in not so long. And we're also now studying, basically, as I was alluding to, trying to understand what are the molecular mechanisms that mediate the survival of clones and the to lead to metastasis at the end of the day, because that's something that is critically what drives basically aggressive disease as well. So understanding what are the drivers of metastasis and how we could intercept them is something that we are investigating at present.
All right. Well, thank you so much, Dr. Cortés-Ciriano, for joining us on OsteoBites today and for making it better for osteosarcoma patients. I think this work is really important and foundational for helping us really think about how to attack it differently as we better understand the biology. So more information on all of this and all OsteoBites can be found on our website at mibagents.org, on the MIB Agents YouTube channel, and at your favorite podcast place. And we hope you can join us next week for our last OsteoBites of 2025. We are going to be joined by Dr. Katie Janeway and Dr. Suzanne Forrest from Dana-Farber Cancer Institute. They will be discussing results from the olaparib and the ceralasertib phase 2 trial in patients with recurrent and unresectable osteosarcoma. You can find our OsteoBites lineup for the next few months on our website. You can subscribe to our YouTube channel or follow us on Spotify so you never miss an episode. And if you have any ideas for other topics that you'd like to hear about, please share them with us at events@mibagents.org. Thank you again so much, Dr. Cortés-Ciriano, for spending time with us today. And thanks to all of you for taking time out of your day to join us. We hope to see you back here on OsteoBites next week on December 11th, when we are joined by Dr. Janeway and Dr. Forrest. Thank you so much. And thanks for staying up late for us, Dr. Cortés.
Thanks, Christina. My pleasure always.
All right. Thanks. Bye-bye.